and I'm happy to present because I've played a scientific advisor role with REPROCELL for many, many years. And what I want to try and focus on today is the stem cells, stem products and services which are available in India to the clients who have been focused on mesenchymal and stem cell iPSC-derived cell work in India. But before I go there, I wanted to give you a couple of pieces of information about the company. So REPROCELL is now not a small company. It's quite a large company with headquarters based in Japan, and it consists of three different units. It has a unit in Europe called REPROCELL Europe. It has a unit in the USA, which is called REPROCELL USA, and it's got BioServe, which is a unit which provides tissue services worldwide. So as you can see, then the company is involved in all aspects of the development cycle, both from sourcing human tissues to providing cells for drug discovery, all the way to doing editing and looking at making iPSC cells, and also providing diagnostic services. I'll only focus on a couple of aspects of this, but those of you who are interested in all these other services that are available should feel free to go to the REPROCELL website and look at the variety of things that are offered. Okay. The business structure of REPROCELL is divided into so the discovery capabilities include human tissue samples and molecular services, iPSC reagents and services, bioengineered tissues and 3D models, and preclinical human fresh tissue sourcing and testing. So on the therapeutic side, the clinical capabilities include using MSC and iPSC-based cells for therapy, including commercial discovery services and commercial therapeutic applications. It has a diagnostic arm which does tissue typing, HLA services, and microsome analytics. There's a precision medicine arm which does preclinical patient stratification using AI and human tissue testing. And then it supplies a variety of clinical reagents, including clinical reagents for MSC and iPSC work. So stem cells, REPROCELL stem cell history, stem cell scientists around the world are using REPROCELL services. And that's because REPROCELL has been the first in many of the things that it has done. In 2005, REPROCELL introduced primate ES medium, which was used in the earliest iPSC studies. In 2009, REPROCELL introduced ReproCardio iPSC-derived cardiomyocytes, and that was the first of its kind product that was introduced. In 2010, StemGen was the first company to commercialize RNA-based reprogramming, and this is now available in India. StemGen was one of the early leaders in providing stem cell-related small molecules. And in 2016, REPROCELL Medical has established a partnership with StemChymal for cell therapy technology. I'm going to try and focus on RNA programming because this is a big part of the services and a unique part of the services provided by REPROCELL. So there are many ways to do non-integrative reprogramming to generate iPSC cells. And perhaps many of you are familiar with Sendai-based reprogramming. However, Sendai-based reprogramming has some advantages and some disadvantages. One of its advantages, of course, is that it's relatively easy to do and you can use it with blood cells. However, you also need methods which don't involve virus persistence, but are still non-integrative and relatively easy to do. And one such strategy has been what REPROCELL provides, and that's a whole kit or reprogramming system for deriving iPSC, which is relatively easy to use. It's the StemGen Kit 3, which is virus-free, DNA-free. There's no virus in it. It's also, because it's an RNA kit, it's integration-free. And importantly, it can be done using a feeder-free culture system, so it can also be clinically compliant, provided you use the right reagents. It's got relatively fast reprogramming kinetics, so you can get colonies within 10 to 15 days, and it's highly efficient, so you can use fewer starting cells, and it has been shown to work with a variety of cell types. The whole reprogramming kit has been patented, which means that you don't have to worry as you move forward, you have a one-stop shop where you can go for licensing requirements. So when you compare these systems, you can see that this programming kit has some advantages compared to both episomal and Sendai virus. It's quite efficient compared even to Sendai virus when it's used for adherent cells, and it's certainly more efficient than episomal cell pre-programming. There's no integration, there's no retention, there's less screening required compared to that required for Sendai virus or for episomal reprogramming, which there's a certain fraction of integration of the plasmid, and the workload is relatively straightforward. And the rate of aneuploidy that you see with mRNA-based reprogramming seems to be less than what you see with episomal and Sendai-based reprogramming. A lot of this has also been published on, and I want to highlight the thing that you're seeing on the right. This was developed with BioNTech. And as you know, BioNTech has been famous because of its collaboration with Pfizer for its mRNA vaccine-based development efforts. And StemGen was able to work with them on the deep knowledge of how to reprogram and how to make RNA less immunogenic. And a lot of that has been published in this paper, and I'd encourage you to look at this if you're interested in looking at mechanisms of reprogramming and how these kits work. Okay. The third gen reprogramming kit what I'm talking about and which is available here in India consists of the standard reprogramming Yamanaka factors. It contains a set of microRNAs, and it contains what are called EKBs, or small molecules, which are peptides, which are interferon escape mRNAs. And there's an optimized liposuction protocol for adherent cells. And there is a feeder-free media with a GMP-compatible system of I-matrix substrate as well. Why do we use these interferon response EKB kit components? And that's shown in the figure that you see here. Each of these affects the immune response or the interferon response to synthetic mRNAs. And this is a really important component in making the kit more effective and also reducing the innate immune response that occurs to this sort of reprogramming, making the iPSC cells you get overall qualitatively and much better than they would otherwise be as compared to the other systems. Okay. The protocol for reprogramming is also relatively straightforward and is detailed in the documents that you will get with the kit. You plate the cells. Because it's feeder-free, you can start the process pretty immediately. There are 4 transfection that you do, and then you form media changes, and then you pick and maintain colonies. Because you don't have to worry about integration artifacts and persistence of virus, it makes the whole protocol and getting iPSC lines relatively short. Okay. The reprogramming kit is also very cost-effective, though I won't talk about that here. And what you see is how the components are provided. You get the mRNA in one tube, you get the EKB and RNA in another tube, you get the microRNAs that are associated with this in a third tube. You have a detailed protocol on how to combine them and at what concentration to combine them. And then you can also buy the xeno-free media that's available, NutriStem, and the I-matrix, which is recombinant protein-derived material that's also available. And you can get a cryopreservation media for your iPSC, which is also animal component free. Although the kit is marketed for research use only, because the patents are available with REPROCELL, because of its acquisition of StemGen, a commercial license is also available. This commercial license allows companies to use it for commercial activity other than for therapeutic use. So you can use it for all purposes other than using these cells for therapy, for which there is a separate protocol and a separate set of kits and reagents available, which are similar to this kit, but further license requirements exist. Not only has StemGen made the kit available, and each kit generally can allow you to reprogram up to 9 different lines or 9 different times. And there are also lines made using this kit which you can purchase. And that's available from many companies. But what I want to identify highlight here is that REPROCELL has made an effort to make iPSC lines from Indians. And so they have several male and female Asian Indian lines made from relatively young donors as well as old donors using skinned fibroblasts as a sample, and that these lines have been fully qualified, tested, and stocks are available off the shelf for use. And these can be used for commercial activity, non-clinical use, licenses for that are available. But if you're using it as an academic and internal research, you just purchase this under the limited use label license that it comes with. This means that you have a very clear path from going with a low cost academic research use product and knowing that the exact same product can be used in a commercial use as you move forward with something exciting that you have developed. I'm going to switch here from the iPSC to products that are available for mesenchymal stem cells. But before I do that, I just want to remind you of the three important points I made. Cells are available, kits are available to make iPSC, and commercial licenses for this are available. And all of this is available directly from the center in Hyderabad, from which you can purchase these. So the timeline and turnaround time for orders is relatively short and straightforward. I'm going to make the same points for MSc and point out which reagents are available and why this has been important. So as you know, MSc is a big group of cells and they can be isolated from multiple sources of tissue, and they are then expanded for use either in research use or under GMP conditions. And then these are used in a variety of different ways. One new way that people have considered using them is to use them as starting material for making exosomes, because exosomes MSc turn out to be a high producer of exosomes. So the products that REPROCELL has spent quite a lot of time working on are the following. One is to make MSc from iPSC. This becomes a renewable source of MSc. And because they already had iPSC lines, they have been able to make multiple cell lines derived MSc. So multiple clinical and non-clinical lines are available. The primary MSc, there is a worldwide distribution of GMP grade bone marrow derived marrow cells. And they are the sole global distributor for these cells. Because they can make GMP grade MSc available, they've also made sure that there's GMP grade media for culture and propagation of these cells for your efforts. That media is a xeno-free media. It's the NutriStem media that they developed, and it comes in a package which contains the main basal media along with a set of supplements. And there's, of course, complete protocols and documentation for regulatory submission, which will be required. So you have documentation related to the iPSC and documentation related to the protocols to make MSc and multiple starting MSc populations for you to test before you decide which iPSC line you may want to use. Okay. GMP grade and research grade MSc and exosomes. So as I pointed out, because you have iPSC lines, which are clinical grade, and because you have a source of GMP grade bone marrow derived mesenchymal cells, you have two different sources of GMP grade MSc, and you have cost-effective research working banks made for use as well. These MSc have been well characterized, and you can see that in the series of slides, and I'm not going to walk you through all of them, but I wanted you to concentrate on the right side of the slide, which talks about the MSc exosomes. So these same MSc, after they've been put in culture, have been tested for their exosome production, the quality of the exosomes, the phenotype of the exosomes, and the ability to be able to use them and what their loading concentration and the marker, exosome markers that are expressed. So you can see the applicable opportunities for these reagents is limitless depending on what academic research you are doing. And what's really important is there's a seamless path going from academic research to clinical applications. Okay. So I want to summarize all of this in one slide and say that not only can you get the reagents for your own use, but you can also get services associated with all of these things. And that's what's summarized in this slide. So you can have custom iPSC generated as a service. You can have custom MSC generated for you as a service. You can have reprogramming from your tissue, or you can get tissue sourced from REPROCELL and have them do the entire process from start to finish. You have a diagnostic piece where you can characterize the cells. You have all the reagents that you need that can be used. And then there are differentiation protocols that, not all, but a significant number, which REPROCELL has mastered, and they are available as a service when you want to move forward. And here's an example of what's the workflow for making cardiomyocytes. Remember, I told you that REPROCELL was one of the first to introduce iPSC-derived cardiomyocytes. So you don't have to sign on with REPROCELL for the entire set of services. You can do it in a modular way. So you can ask simply for tissue procurement, or simply for cell line establishment, or simply for differentiation. So it's modular and it's customizable. Yeah. So some highlights from the differentiated cells that REPROCELL has internal service expertise in and has provided to a variety of clients includes making neurons, including characterizing them with MEA and patch clamping, looking at dopamine release and reuptake through synaptic vesicles. And there are custom media which are required for these assays, and all of those are available from REPROCELL. Stem RNA sensory neurons are also another product that's been introduced by REPROCELL. These are iPSC-derived sensory neurons, which can be shown to be true sensory neurons in the sense that they respond to characteristic or well-known stimuli required by sensory neurons, including response to capsaicin, menthol, temperature, etc. They express the appropriate sensory neuron markers, and they're suitable for disease modeling. And they, more importantly, they express the characteristic pain channel TRP5 and TRP6. Custom lots of these cells are available. Another piece that's available is a mixed population of cortical neurons. These are also iPSC derived. They mimic the in vivo population, and they have been used in vitro in drug discovery purposes, and they've also been transplanted in vivo. And the activities are consistent with member subpopulations that have been identified in the CNS of cortical neurons. But this, I want to emphasize, is a mixed population of neurons. So this slide I just want is here just as a placeholder to remind me to emphasize that these products for stem cells are now stocked and delivered from the Hyderabad facility that Stempeutics REPROCELL has identified, not Stempeutics, sorry, REPROCELL has established in Hyderabad. I'm now going to go forward to one more piece, which I think is really important in terms of differentiation of the cells. And REPROCELL has made a lot of effort in looking at these signaling pathways and has identified small molecule signals that are useful in directing differentiation along particular lineage pathways. And that's sort of listed here, as you can see. This includes the TGF-beta BMP pathway, the Wnt pathway, the hedgehog pathway, the FGF pathway, and of course, integrin signaling. And you are all probably familiar with many of these factors and markers that have been used in directing differentiation. The importance of emphasizing this list is that REPROCELL has carefully curated these factors and calls them stem molecules, and these are available from REPROCELL either already solubilized, which is often a problem when you're doing this, pH balanced so that they don't affect your cultures, and the quality has been much better than what you can get from many of their competitors. Okay, so it's a USP grade set of a small molecule portfolio for stem cell research. It affects all the known major differentiation pathways. These are much more cost effective. And as I pointed out, they're available at high purity, and they're available as solubilized factors as well. So again, the importance of this list is to highlight some of the ones which are available and in stock and available ex Hyderabad. So if I can summarize the stem molecules, It's a comprehensive portfolio. Each lot is tested for high purity, sterility, and toxicity, and both bulk and custom sizes are available. So as with all products that ReproCell tries to sell, there's a seamless transition from academic to translational to clinical work. The same idea has been true for growth factors and cytokines, and these are some of the growth factors and cytokines that are available. So ReproCell is not a one-stop shop for all cytokines that you want to get, but cytokines related to work that's being done with iPSC and MSC and their differentiation is, of course, available from ReproCell as a tightly focused set of things so that you can get all the reagents you need for these two cell types and their differentiation into neurons and cardiomyocytes from one single supplier. This includes not just mRNA molecules, it does not just include small molecules, and does not just include the factors that we talked about, but also includes media, which I've provided you with some mention earlier. So the stem cell media that's available is actually made in partnership with Sartorius, and that includes the NutriStem set of media for which ReproCell is a distributor. In India, it has xeno-free media, which is ideal for growth of iPSC and ESC. It's really very competitive in its pricing, and the expansion rates that you see with this media are equal to or better than most of the other media that's available. It's manufactured under GMP conditions, and all the media has its own drug master file. Again, to make the same point that I've made several times today, is that it provides a seamless translation from the research grade to the clinical grade path that you might want to walk along. The MSC media is also called NutriStem. It's also xeno-free media. It's MSC NutriStem. The iPSC media is an iPSC NutriStem. There's a freezing media which goes with this, which has been matched so that the cells grown in this media can be easily cryopreserved in the cryopreservation media. And as I told you, there's a lot of work which is being done with primate ES cells, non-human primate ES cells, and that's a great media for feeder-dependent cultures that people still perform there. Okay. The last piece that I'm going to talk about is stem cells for clinical use. And as I pointed out, that there's a seamless path on reagents, media, and substrate. What about the cells themselves? And ReproCell initially made a promise that they would make a large number of cells and make sure that they'd be as close to clinically compliant as possible, and then took the next leap to ensure that there were GMP manufacturing facilities and that they could also be your partner for manufacturing cGMP, iPSC, master cell banks, and seed stock. And this is done in either the USA facilities or it's done in the Japan facilities. And so the entire workflow of making a GMP iPSC line is not as straightforward as you would think. It includes making sure you screen the donors appropriately and not just screen for that particular geography, because your plan may be to use them internationally or globally. And so you have to make sure that the donor screening is compliant with all the different regulatory authorities. You need to acquire that tissue after you have screened the appropriate donor. If you're doing the reprogramming, you want to make sure that that's appropriate documentation and freedom to operate. You need to make a seed stock of those cells and maybe make a decision on whether you're using a polyclonal population or a monoclonal population. And then you need to look at whether you have clinically compliant iPSC clones, which are karyotypically normal. And that's an extensive set of characterization done there. You then choose a clone which can be evaluated by you to be able to understand whether it works well in your manufacturing process. And should you have that, you want to be able to make both a master cell bank and a working cell bank for your routine use as you establish your manufacturing protocol for clinical grade differentiation. ReproCell aims to be your partner for this entire process. And that's what you see here. And what you also see is the timeline required for what needs to be done. And so keep this in mind. If you need a clinical grade working cell bank and a master cell bank, or before you get to differentiation, the entire process will take you about 18 months. And it's generally a little bit longer, not because of ReproCell, but because your needs or requirements or the testing may change. So I'm not going to walk you through all of these slides, but this is what really each of these steps can be broken down into, right? Tissue acquisition sounds like a nice one-step process, but it really means getting the skin cells, making sure there's no contamination, establishing the fibroblasts, making sure that you get reprogramming and that's done, and that you have a backup in case it fails the first time. You need to make sure that you have an appropriate place to store your seed stock while you're doing your quality control checks. You need to then be able to expand your seed stock, make sure that you've done the appropriate quality control checks before you can even start making your master cell bank. Right. Now, how do you access this whole process? Remember, I started off by telling you that when we were talking about other cells in terms of services that you can enter into it at any module. I want ReproCell has used this exact same concept for what needs to be done for clinical grade cells as well, right? You can either get the kit, you can get the fibroblasts, you can get the seed clones, you can get the differentiation process, you can get a master cell bank. And so that's a really important piece to keep in mind as you consider who you want to use to be your partner in your iPSC journey. So custom seed clones prepared to specification of the sponsor, including donor specifications if you need them. Ready-to-use seed clone banks are available, and pilot clones of pre-made master cell banks are available for your testing. This is the sites where they are made. In Japan, there's an operational GMP facility, and the USA existing clones generated are expanded. And this is using what's called GMP in a box system. It's the biospheric system. It's a closed GMP system, which is now available in the Maryland site in USA. The last piece, which has become exciting for people, and I really want to emphasize this really quickly, is that people have realized that allogenic iPSC cells and their differentiated products need to be modified further because otherwise they will be rejected, much like any other cell or tissue is rejected because of their HLA mismatch. So now there are multiple ways of making universal cells, but the starting material for a large variety of methods of making universal cells is to make a class 1 and class 2 null cell. So that's like the baseline class 1, class 2 null, to which you add additional modifications to make a universal hypoimmune or universal cell. And because you always need to test whether any of these modifications affect the differentiation process or manufacturing, ReproCell has become one of the first companies to offer research-grade class 1, class 2 double knockout iPSC cells and iPSC-derived MSC for use and evaluation. The way this was done was using CRISPR-Cas9. This was done by the Japanese main headquarters in this non-GMP facility, as these are research grade, though they have the ability to make it in the research facility as well. And then they did it in a sequential manner. They knocked out B2M first, and they knocked out CT2A next. And they were able to show that they had good quality iPSC that were karyotypically normal, and they had the ability to differentiate into many different phenotypes. Remember, this doesn't mean you have to use only this line. What it means that as a service, you could have your own favorite line or custom line and have as a service edited by using CRISPR-Cas9 to make class 1 and class 2 null cells. I won't go through the quality control, but they went through the same rigorous quality control and their ability to not or fail to express HLA, and by sequencing to see whether the knockout had actually occurred and it was homozygous or heterozygous at its site, and that these cells also went through the same standard quality control assessment that's done for all iPSC, so that you get a full certificate of analysis when you get these cells for evaluation. So I'm going to end here, but before I end, I want to try and summarize what I try to tell you in as simple process as possible. And so I'll give you the 4 take-homes that I have for this whole system. So you have a one-stop shop for which is very focused in providing you a seamless transition from the academic side through a translational side to a clinical side. And it's important to keep in mind that expenses go higher. And so what ReproCell has done is to make sure that you have an ability to enter and choose what stage that's appropriate depending on the stage your internal research work is at. And then to make sure that you don't fall into the trap that we often find in India where things are back ordered or because they have to be shipped, there are delays or there are clearance issues. What ReproCell has done is made an effort to set up a facility and a warehouse here which can be used for shipments so that you have fewer back order and shipping issues. Here is the contact information for all of that. You already heard Sunil introduce himself, and these are the contact sites and websites that you can use to be able to place your orders. If you have any custom service questions, you can contact them directly. I'll end here and I'll be happy to take any questions.
Uh, Thank you, Dr. Rao, for this exceptional presentation. So we got some queries from one of the participants. So here is the first question. So the client, I mean, do the client need to provide their own cells or REPROCELL can program them?
Yeah, it's both ways, right? As you know, from ReproCell, and as I try to emphasize here, you
Mhm.
can send your own cells, but remember, it may be an additional charge if you send your own cells because you cannot accept the cells into a GMP suite or work with them without doing a series of tests. And so those tests will have to be done, and your cells will be in quarantine till that's been tested. Once that's been tested, then the process is exactly the same. Of course, if you use ReproCell tissue to do reprogramming, then all of that's already been done, so they can start immediately. And for editing, you can send your own iPSC line, and the editing can be done in that line. It doesn't have to be that you have to take a ReproCell line.
Okay. There's one more question, like how are the MSc reprogramming from the IPSCs? So this is one question from one of the participants.
So, yeah. So we use a differentiation protocol which has been developed. It's not a patented protocol. There are 2 different pathways by which MSC can be made, or mesodermal or mesenchymal stem cells can be made. One is what's called the neural pathway, where you make neural crest, and neural crest makes mesenchymal stem cells. And the other is the mesodermal pathway, in which you go through and make intermediate mesoderm, from which you can make muscle or you can make connective tissue, and that's the connective tissue in which you get mesenchymal stem cells and bone marrow derived hematopoietic stem cells. So ReproCell uses the second pathway and has been able to make mesenchymal stem cells using a standardized differentiation protocol, which covers about 28 days.
Okay. Here's another question, do the client need to pay the license fee for each line they purchase, and what could be the passage number for these IPSs?
Yeah, so the license fee is per line. For non-commercial use, there's a different price. For clinical use, it is
Right.
a different line made with a slightly different protocol, and there's a license fee associated with that. All I can say, because I'm not directly involved with ReproCell, I'm an advisor to the company, is that, but what I do know is that the prices for licenses have started coming down as the ability to make clinical grade iPSC lines has expanded, and the ability of, or number of lines that are available for licensing has increased for non-therapeutic use as well, that license prices are come down. I have found that ReproCell license prices are quite competitive, and they have a very clear-cut model, which is important. They provide cells for evaluation for a minimal price so that you're not committed to buying an iPSC line, which you may or may not work in your system. And then once you've gone through the evaluation process, there is a license fee, but that license fee is also, unlike others, there's no maintenance fee. It's a single one-stop fee with a minimum royalty payment at the other side. So that structure has become very attractive for people who want to work with ReproCell.
Okay. One last question. There's a question on the efficiency rate of the STEM and reprogramming kit. So what is the efficiency rate of this kit?
So the efficiency rate is a little bit variable because often it depends a little bit on the users. No matter how much you standardize a kit, there's a little bit of experience and etc. that's required. But stem cell generation 3 kits are really very efficient. So I have used like 500,000 cells or 1 million cells and gotten hundreds of clones rather than the tens of clones that I get with a lot of other systems.
Okay. So, thanks, Dr. Mahendra Rao. I think like most of the questions, rest of the questions are related to the costing. So I think like you'll just, I mean, just they can just drop us. But anyways, thank you all for joining us today for this insightful webinar. And a special thanks to Dr. Mahendra Rao for sharing this thing with the latest advancement on the stem cell. And like if you have any further questions, like we would like to learn about the referential offerings, please feel free to reach out. Like the contact details are available.